Y bueno, les presento a nuestro primer ponente, el doctor Christian Oberkanins. Él es director de tecnología en Vienna en Austria. Él es químico de formación por la Universidad Tecnológica de Viena en Austria. Después también ahí en Austria, en la Universidad de Viena, él obtuvo su doctorado. Tiene alrededor de 65 publicaciones científicas en su haber. Más de 200 contribuciones en congresos nacionales e internacionales, tanto orales como en póster. También él es desarrollador de 2 patentes de sondas métodos y sondas para el diagnóstico genético de la hemocromatosis y métodos y sondas para el diagnóstico genético de la hemocromatosis hereditaria. Entonces, el doctor Christian ha participado con nosotros en este tipo de sesiones de las jornadas científicas durante los 3 últimos años, dando diferentes pláticas. Estamos muy contentos de que nos acompañe de nuevo este año, así que le damos la bienvenida y le voy a ceder el micrófono y la pantalla. Dr. Christian, I have told the attendees that you have participated with us in these sessions for the last 3 years, giving different talks. We are very happy that you will join us again this year. So we welcome you back, and I give you the screen and the microphone for your talk. Welcome.
Thank you very much, and a warm welcome to everyone here from Vienna. It's a pleasure to participate in your conference again, and I'll be very glad to take the opportunity to give you an introduction to what Vienna Lab did over the last couple of years in next generation sequencing, and especially in the sequencing of the human microbiome. Let me start by introducing everyone who is not yet familiar with Vienna Lab, giving you an introduction to what Vienna Lab is and what we do. So the company exists since about 30 years, was introduced here as a spin-off of the Medical University of Vienna, and is developing and producing CE-marked IVD in vitro diagnostics. Vienna Lab is distributing the product in collaboration with dedicated and experienced local partners in about 60 countries worldwide. And we are very happy that Uniparts is taking care of our products in Mexico. Our target customers are hospitals, diagnostic laboratories, universities, and pharma companies. And Vienna Lab works under the ISO 13485 quality management system. We currently deal with 4 or 5 areas of human disorders. The first group is hereditary disorders, so mutations that directly cause a disease, a disorder. The second one is genetic predisposition, so mutations that confer an increased or decreased risk to develop a disease. The third group is pharmacogenetics. It's inherited mutation that determine the response to therapy, like resistance, side effects, optimal dosing of a drug. The fourth one is oncology. This is somatic or acquired mutations that cause tumors or determine the response to anti-cancer therapy. And the fifth group, which will be the main topic of today, is microbiology and infectious diseases. We introduced last year a real-time PCR test for SARS coronavirus, for example, and we use next-generation sequencing to determine the microbiome in the gut or in the skin of patients or of subjects. This is the 3 technologies we use at Vienna Lab, depending on the complexity. If we deal with a few markers, like 1 or 2 markers only, we use real-time PCR. We use Tuckman technology, and we call the products the real fast assays. If we deal with a medium complexity between 3 and 40 mutations or SNPs, polymorphisms, we use a strip assay. That's PCR combined with test strips. And the test strips produce visible results that can be read like a barcode to see if a mutation is present or not. And the third one is for high complexity questions or topics with more than 40 markers, polymorphism, mutations. And in this case, we use next generation sequencing, or NGS. Today, I want to entirely dedicate the time to next generation sequencing and the human microbiome. To start with, I want to briefly give a history of the sequencing technology to see where we stand. DNA sequencing was originally developed by Frederick Sanger in the 1970s. The so-called Sanger or the dideoxy sequencing became very popular, initially done with radioactive labels and with X-ray films, later with fluorescently labeled markers and instrumentation. And this was using dideoxynucleotides, the termination chemistry, and produced 500 to 1,000 base pair fragments. And this technology was used extensively and exclusively for about 30 years in all labs worldwide. Around 2005, the so-called second or next generation sequencing technology became established and spread. The first commercial instrument that became available was the Roche 454, and later on this became replaced by instrument mainly provided by Ion Torrent, now part of Thermo, or by Illumina. And this is what still is the most popular way of let's say modern sequencing, and collectively and generally is called next generation sequencing, or NGS. So this developed around 2005. And the next step was taken a couple of years later. Around 2011, PacBio developed the long read sequencing, and Oxford Nanopore developed the nanopore sequencing. This is still developing technology and not yet as widespread in the labs as the Illumina or Ion Torrent technology, but in general, this is called the third generation. I want to focus on the second or next generation sequencing because this is what we use and what many labs use nowadays. The basic NGS workflow starts by from a DNA sample with a library preparation, followed by target enrichment. So a certain part of the DNA is taken and amplified or selected out of a pool. Then there is the sequencing, mainly limited to Illumina or Ion Torrent technology. And finally, because all of these technologies used so-called massively parallel sequencing, the enormous amount of data have to be analyzed using computerized tool. This can be off-the-shelf software or can be customized software that is linked to a certain application or kit. Usually the library preparation chemistry or technology is linked to the type of sequencer. So if a laboratory is equipped with an Illumina sequencer, it has to use another library preparation technology than a lab that is equipped with Ion Torrent technology. There are two major target enrichment strategies used. The first one is called amplicon-based assay or technology. Essentially, this is PCR-based. So tiled amplicons are generated that cover the whole area of interest, and then these amplicons are sequenced, and the information from this sequencing is combined and analyzed by a software, is aligned to give a whole sequence information finally. The second one is hybridization capture based assay or hybrid capture based. This uses usually magnetic beads or beads that are linked to probes, and these probes bind to the sequence of interest and kind of fish out the sequence of interest, and then only these sequences are only these fragments are sequenced. The amplicon-based technology is much easier and quicker, while the hybrid capture-based technology is more sensitive, so it's very often used for oncology application or for large regions of interest. The Vienna Lab NGS tool for the microbiome is based on the amplicon technology, amplicon-based assay. This is a brief overview of the sequencing workflow. So the library preparation is the first step. In this case, we offer the tools for that. So the DNA is prepared, adapters and indices are ligated, and this is everything included in the kit that we offer to the lab. Then there is the sequencing parts that are taken by tools that are offered by Illumina. So there's a cluster generation. The clusters are arranged in the flow cells, and then the sequencing reaction is run within the instrument. And finally, the sequencing information that is collected is analyzed by a bioinformatics tool that is again offered by Vienna Lab. The second part of information or introduction I want to give today is about the microbiome. So what is the microbiome? The name comes from the Greek language and combines the term micro, which means small, and bios, which means life. So it's small type of life, microorganisms. It contains bacteria, archaea, fungi, algae, protists. Usually also the viruses are called as part of the so-called microbiota or microbial community. I put a question mark there because sometimes it's also this part is more included within the so-called theater of activity. What is called the theater of activity refers to the entire habitat, the so-called biome, with its microscopic organisms, so the microbiota, plus the genomic information, the surrounding environmental conditions, the metabolic activities, the ecologic functions. So all that taken together is the so-called microbiome. The microbiome inhabits nearly all parts of the human body and also, of course, of our environment. So also, of course, you can take microbiotic samples from the soil or everything around us. But the human body, the part of the human body that are mostly interesting for microbiome analysis is the nose, the mouth, the lungs, the stomach or gut, that's called the intestinal or gut microbiome, and that I want to focus today, the colon, the sexual organs, and the skin. What influences the microbiome? It's of course genetics, but also the environment, the diet, the lifestyle, hormones taken, industrial products. So a lot of things contribute to the microbiome and can influence the microbiome and also over time change the microbiome, of course. To give you two numbers that for me are, at least are very interesting, the number of microbial cells is estimated to be roughly 1.5 to 10 times the number of human body cells. So everyone contains up to 10 times more bacterial or microbial cell than one has human cells. And the microbiome in total, of course, because it's very small cells, but the microbiome in total is estimated to take up about 1 to 3% of the human body mass or weight. The human intestinal or gut microbiome consists of bacteria, archaea, viruses, and eukaryotes, and it significantly impacts the host immune system and the metabolism. Reduced activity or imbalances, disbalances in the microbiome have been associated with a variety of disease phenotypes, and this is actually science that started only, I would say, the last 10 to 15 years, and the information collected on that, so the influence between, or the association between microbiome and disease is a rapidly advancing field. Very well-known associations are between the microbiome and inflammatory bowel disease, type 2 diabetes, obesity, fatty liver disease, a couple of skin disorders such as acne, eczema, but also neurological disorders. It's known, for example, that the maternal microbiome may influence autism in children, in newborns, but also microbiome can be linked to multiple sclerosis, to depression, and other neurological or psychiatrical disorders. Various types of cancers may be caused or influenced by the microbiome, and numerous other disorders are currently under investigation. The microbiome analysis approaches include either shotgun metagenomic analysis. This is a very complex and very comprehensive, but also very time-consuming and expensive approach, but also microbial metatranscriptome sequencing, microbial whole genome sequencing, and bacterial 16S ribosomal RNA analysis. And that's actually what we are focusing on, because it's highly multiplexable, it's most cost-effective, and for the human intestine or gut, it collects most of the information that is really necessary or of interest to specify the bacteria that inhabit the human gut. What is the 16S ribosomal RNA? It's an approximately 1.5 kb stretch of ribosomal RNA that occurs in all bacteria. It's highly conserved and highly specific. It contains 9 hypervariable regions that allow it and make it a good candidate for taxonomic classification of bacteria and archaea. It encodes ribosomal RNA that is essential for the synthesis of all prokaryotic proteins. You see here a graphic showing the 9 variable regions. They are interspersed by conserved or constant regions. In the graphical depiction below, you see the gray area, which, for example, is identical for all the Escherichia strains. But you see also the yellow part, which is highly diverse even between the strains of the same species. We focus on 2 of these variable regions that are V3 and V4. They are among the most variable and most suitable parts of this 16S ribosomal RNA gene to use for NGS sequencing. Vienna Lab offers the so-called 16S microbiome NGS assay kit. It enables a species-level taxonomic classification for more than 99% of the bacterial communities that usually inhabit the human gut. It identifies imbalances in the gut microbiome and pathogenic microorganisms. It is based on next-generation sequencing of the highly variable V3 and V4 regions of bacterial 16S RNA. It produces sequencing-ready NGS libraries in less than 4 hours. We use dual indexing to pool up to 96 samples per run. It is compatible with all Illumina next-generation sequencing platforms, and the kit, the tool, includes access to a proprietary bioinformatic analysis pipeline. This is the sequence of the protocol depicted here. So samples are collected. That's usually stool fecal samples. DNA is isolated from the sample. We run a first set of PCR amplification to amplify the target region, V3 and V4. Then a sequencing is run of the target regions, and then by using the indexes, the barcodes, the different samples can be demultiplexed, and the software can produce an analysis that identifies the different species or down to species, so kingdom, phyla, class, order, family, genus, and species level information on the composition of the human gut. This is the protocol in more detail. So there is a library preparation part 1, which is which amplifies the target region. It's the target region V3 and V4 from DNA, from extracted DNA. An index primer binding site is added on both sides. So after this first round of PCR, there is a cleanup. You see here the components or the parts labeled blue are additional reagents that are required and not offered in the kit. But the orange part is reagents that are all included in the kit. So the PCR tools are all part of the kit. So the cleanup after the first PCR, and there is an optional fragment size check. And then there is a second PCR, which is used for the indexing. So to be able then to combine all samples into one sequencing run. Also the P5 and P7 flow cell adapters are added. Then there is another cleanup, a fragment size check, and then the libraries must be quantified, normalized, pooled, and then they go into the Illumina sequencer. This is the kit, how it comes to the lab. It contains all the master mixes for the PCR, the forward primers for the first round of PCR, the forward and reverse primers for the first round of PCR, for the second round of PCR, the magnetic beads for the cleanup. It includes an activation code for the software to be used. And in order to allow more than 96 samples to be processed, it comes in 3 different kit formats, reference numbers, set A, set B, and set C. All of them are suitable and have indexes for 96 libraries. This is the instruments and additional materials and reagents that are required. So for DNA extraction, a stool DNA isolation kit is required. We recommend the kit from Norgen Biotech. PCR equipment, of course, a thermocycler, suitable plasticware, nuclease-free PCR-grade water. For sample purification, 80% ethanol and a magnetic separator is required. The library QC, which is optimal, can be either done by agarose gel electrophoresis or by an automated capillary electrophoresis instrument, such as the fragment analyzer or a tape station. For library quantification, one should use a qPCR library quantification kit. We can give recommendations on that, or a qPCR or the qubit the fluorescent quantification tool by thermo. For sequencing, Illumina instrumentation is required. We recommend, or we have the kit is optimized for the MySeq instrument, and we use the V3 600 cycle chemistry to run the kit. The preparation for sequencing, qPCR is recommended, or the qubit. The reagents are not included. We offer on our website a so-called Microbe Calc pooling calculator that is kind of an Excel sheet that can be used to calculate or to assist in the dilution of libraries to the recommended concentration of 4 nanomole and to pool for the sequencing. The second tool that is offered on our website is the Microbe Calculator, coverage calculator, to assess how many samples can be sequenced with the intended sequencing setup. And the third tool is the sample sheet generator. This is the pooling calculator. You see the web address, vienalab.com support, where this tool can be accessed. The user has to input the desired final pooled library concentration. The recommended one is 4 nanomolar, and the desired library volume, which usually is 100 microliters. And then this sheet automatically calculates from the library concentration that has been determined, automatically calculates the pipetting volumes that need to be used for the pooling. The second tool is the coverage calculator. In this case, so the user has to input the desired coverage. In this case, it's 100,000 X. The minimum we recommend is 50,000 X. And you see with the different instruments like the MiSeq or the MiniSeq and the different chemistries that are available, you see how many libraries can be sequenced in one run. And the third tool, also offered on the same website page, is the sample sheet generator. This is something that is to be used when starting the run on the Illumina sequencer. The sample information is directly imported from the pooling sheet, and in this case, the different indices or barcodes have to be and the input of the user is the different index sequences or barcodes, so that the samples, the different libraries, can finally be identified. The next step, or the final step of the analysis, is the bioinformatics. And in this case, again, the tool is provided by Vienalab. It comes for free when the kit is purchased. So it's a code comes with a kit, included in the kit. The software tool uses a couple of external pipeline tools. The read classification is done by Clark. The sequence database used is the Silva. The species taxonomy is done on NCBI. The diversity statistics are done using Mothur, and the interactive charts are provided by Krona. So when starting, the user, so when first time accessing the tool, which is a server-based or a cloud-based tool, provided at the address that is given here, microbiome.vienalab.com. The first thing that the user has to do is sign up, providing some basic information. And for this, an activation code that comes with the kit has to be used. So the user can, as soon as the user has purchased the kit and has obtained an activation code, they can go to the website and sign up. They provide the email address, they have to define a password, and they give the activation code. They have to agree to the terms of services, and then they can start. This is the starting page. Of course, upon first login, there is no samples given. So in this case, I already have included some samples in my personal account. But this is the default starting page. The user can also go to a help tool. You see here, the symbols are marked here. The help tool is a quick guide information of how to navigate our tool. It also provides the contact details of Vienalab so that we can be directly contacted in case of problems that cannot be resolved through the help page. There is also a brief description about the bioinformatics tool and the tools that are used for the analysis. Some literature reference, for example, about the Clark systems are provided. And we provide the terms of service. The user can change the password. It shows the account balance. So when the kit arrives, there is an account of 96 activation codes. One is used for, or the first one is required for sign up, but then there is another 96 samples that are open to be analyzed. When a new kit is purchased, the user can redeem the new activation code, just typing in the new activation code information and redeem. So this is like the actual balance, how many unused samples the user has still in his account. Then there is the upload side. The FASTQ files can be selected and uploaded from the computer, and you have to have an active credit. So when using the upload tool, FASTQ files or FASTQ GZ files can be selected and uploaded from the computer, the local computer. The sample name can be selected. Important here, because it's a server or cloud-based tool, the user should avoid any terms that could lead to an identification of patient-specific data. So some anonymous file name should be selected, nothing related to patient information. Then the upload is started. The user has to confirm that one sample is deducted from their account. Usually paired end sequences are uploaded. That has to be clicked here. You see the paired input file is ticked, and then the upload is started. The upload must not be interrupted, so that the browser window must not be closed or some redirection or reloading started. Otherwise, the activation code is lost. So this, it needs a few, let's say a minute or two. It's very quick if the computer connection is reasonably fast. It's very quickly uploaded and must not be interrupted. Then the files are uploaded, and on the lower right you see they are processed. So when they are uploaded, the software immediately starts to process the files. This is something that also takes a couple of minutes, and then the sample appears in the sample overview, and then the user can start to analyze the new sample. So this is a screenshot from the computer window. You see the sample information is on top, sample ID, the date of analysis, the submitter. Some additional information may be included here. For example, it may be stated whether this was a certain healthy patient, a patient with a certain disorder. Anything that is connected to the sample can be written here. Then, because we deal here with intestinal microbiome analysis, some typical parameters that relate to intestinal or gut microbiome are given. For example, diversity index. The most common one is the so-called Shannon index. You see here, Shannon index for this sample, for example, is 3.6, which is roughly a medium diversity. So you see here both the number provided, but also you see here a graphical illustration with colors. The red one would be a low value. The green one is the optimal value. You see this sample is in the green area, so it's between 3 and 4, which is an optimum or a medium from 4, starting an optimum diversity situation. You can also click here, show more. In case of show more, other index parameters can be selected, like ACE and others. Then there is the Firmicutes to Bacteroidetes ratio, or F/B. In this case, it's 0.6. It's rather low, but is within the balanced microbiome composition. This is something very typical given for intestinal microbiome. Excuse me, let me go back. The third type of information provided is the enterotype. Usually the scientific community discriminates between 3 different ones. One is the typical Bacteroidetes enterotype. The second one is the so-called Prevotella enterotype. And the third one is the so-called Ruminococcus enterotype. So this classification is also done by the software. And then you see here the species abundance table. So you see all the different bacterial species listed in a table with the taxonomy ID, with the complete lineage information. You see the number of reads that refer to a certain species, and you see the proportion of these species within the sample. There is an option of depicting all this in graphical terms, but also to download the different information. So this species table, for example, can be downloaded as a CSV file, can be depicted in a so-called sunburst or corona diagram, and this is interactive, so the user can even highlight certain parts of this diagram and kind of play around or go into more details in investigating this sample. The genus and species level composition can be depicted in this pie chart diagram format. So you see here all the different genus or species in the sample depicted in the format of a pie chart, but also in colored and giving with a percentage rate. The same is true for the phyla, class, order, and family level composition. And you see here the reference rate. So if you go to, in this case, you go to the phyla, you can see here the different phyla listed with the value, the percentage value in the sample and in relation to the reference range, and very easily can be recognized whether it's in a normal range or whether it's in an abnormal range. And finally, a report can be generated, a PDF report that is also customizable. You see here this PDF report lists the Vienna Lab tool, so this is the NGS 16S microbiome kit. It gives the sample ID, some information about the sequencing run, how many reads, the percentage of reads that could be identified, the F/B ratio, the enterotype of the sample, the microbiome diversity, like the Shannon index, the phyla, the classification of the genus and species level composition, and the phyla, class, order, and family level composition. And on the last page, it lists the most abundant species. In order to keep the report to a useful format and to a useful number of pages, the cutoff from this table is only species that are more abundant than 0.25% are listed here, but all the ones that are less abundant are, of course, listed within the program. So even the very low abundant species can be identified with our web tool. Finally, to summarize the key features of the 16S Microbiome NGS assay, it's a targeted library preparation kit plus a bioinformatics analysis and reporting tool. It offers a species-level classification of bacteria in the human gut. The time from extracted bacterial DNA to ready-to-sequence libraries is less than 4 hours. Dual index libraries allow for a large-scale pooling, so up to 96 samples can be included per sequencing run. And the kit is compatible with the Illumina sequencing platform. MiSeq, the MiniSeq, even the iSeq can be used, of course, with different number of samples depending on the size of the instrument. So that was my input. Thank you very much for attending, and I'll be happy to take questions and answer them as well as possible. Thank you very much.
Thank you, Dr. Christian. We have a few questions here. I don't know if you are listening to me.
Yes, I'm listening.
Okay. We have a few questions for you. Alejandra Reyes is asking you if there is a correlation between a low intestinal microbiota within the stomach, and if this low intestinal microbiota is correlated with stomach, colon, or intestinal cancer, considering the relationship between the microbiota and gastrointestinal diseases.
Definitely there is. It's not the low number, it's mainly the variety. So the higher the variety is, usually the more healthy the intestinal microbiome is, and the benefit to the patient. But there is definitely a correlation of certain types of bacterial composition with irritable bowel disease, and of course, as a consequence, also with stomach or colon cancer. So definitely there is, yeah. This is something that literature is accumulating very much over time. And of course, this is the most direct, the most clear relationship. For me, the most interesting relationships and unexpected relationships is, for example, also with neurological disorders, with psychiatric disorders, and this sort of disease association is accumulated very rapidly, and there is very good information everywhere to be found on the internet. Even Wikipedia nowadays offers a very comprehensive and good summary of the disease association with microbiome composition and diversity.
Okay, thank you, Christian. We have another question from Enrique Fidalgo. Is this assay quantitative or just qualitative?
It's qualitative and quantitative, so it offers an overview over the different bacterial species that are present in the gut, but it also gives the percentage and the relationship between the different species. As I said, the variety is, for example, depicted, so how many different species are represented in your gut, but also how many percent of your bacterial composition is occupied by a certain bacterial species. So it's quantitative as well.
Okay, thank you. Eric Bautista is asking you, is this assay can be applied to marine animals, or the platform is only for humans?
I don't know enough about the marine species. You mean if environmental samples can be taken, of course, what the tool does, or what the kit does, is analyzing the 16S RNA of bacteria and archaea and giving the information what sort of bacteria is present in a sample. So in principle, you can take any sample. You can take a piece of soil and you can analyze which bacteria are present in this sample. But the software tool is more dedicated to the human intestinal. So this sort of variety coefficients or enterotypes, this is specific information that makes sense for human intestine. And of course, that's what the kit was developed for. But in principle, although it's not dedicated for that, you can take any, as I said, environmental sample, definitely animal sample, and just ignore some of the parameters that would be human intestinal specific. So the enterotype gives no useful information for soil samples, for example. But you can ignore it and just take the species information as an output of the software as well.
Okay, thank you. Another question is, how helpful is this assay to diagnose some consequential diseases like obesity, inflammation, diabetes, liver diseases?
I think this sort of the acceptance in the scientific and medical community is, as I said, is quickly arising. So if you would have asked people 10 years ago, no one would have taken attention of the microbiome. Within the last years, both the information that is available on the microbiome, but also the attention that it gets is quickly increasing. And now there is even as a kind of advanced stage of the original Human Genome Project, which identified all the genes in a human, which was finalized about 20 years ago already. There is a Human Microbiome Project. So a Human Microbiome Diversity Project that is a worldwide project that now dedicates resources to identifying which sort of microbiota are present typically in the human or on the human. So the gut and intestinal microbiome is most important and takes most of the attention. Also, for example, the mouth microbiome or the vaginal microbiome, but some others like lung microbiome and things like that are poorly investigated still. And the disease association as a consequence is quickly establishing. So I think in a couple of years, everyone is very well aware that certain types of cancer are for example, caused by a aberrant microbiome or skin irritations, but also because these bacteria, for example, they create something hormone or substances in the gut that are similar to hormones. And these hormones, for example, might even direct your mood. So depression might come out of some disturbances of the microbiome. And this sort of information is quickly accumulating and will definitely take much more attention in the coming years in medicine.
Okay, Christian. And the last question, how difficult is for a technician to jump from traditional PCR to NGS technique for this kind of assays, including the software management?
The most important thing is, of course, to invest into a sequencer and to have the NGS equipment from Illumina available in the lab. All the other things are practically offered and included in the kit. So what the user gets is a kit that allows them to run the PCR, to take the purification steps, and finally a software tool that is, as you might have seen in the screenshots, that is very easy to navigate. So even someone that has no bioinformatics education or know-how per se can use this tool without any problems. So the biggest step, like to switch from PCR to the NGS tool, is to purchase the Illumina equipment. And this can, as I said, you can even start with the iSeq. The smallest Illumina instrument is suitable to run the ViennaLab tool. Of course, you cannot multiplex 96 samples. But if you have a very low number of samples and your turnover is not yet useful for a MySeq, then you can start with the lowest investment and purchase the iSeq or the MySeq and start from that. So that the know-how is no further know-how is required to use the ViennaLab tools.
Okay, and we have a last question. Talking about the results, the output files, Eric Bautista is asking you, can I get the files with the Otus results to analyze them in other programs, for example, R or Primer?
The software can export, so the import to the software is FASTQ files, so the sequencing output, which is generated by Illumina. This you upload into the ViennaLab software tool, and you can export the species table. This can be exported as a tab delineated files, as a CSV file, exported and, for example, processed in Excel or in any other suitable tool. Maybe, I'm not sure if that answers the question. So the input to the software is the sequencing file, and the output it is a CSV file or a PDF generated report.
Yes, I think that answered the question. Well, we don't have any more questions, Christian. So we thank you very much for your presentation today.
Thank you very much for giving me the opportunity.
This topic has generated interest among the attendees, and we thank you for your willingness and time. And we send you so much greetings from Mexico.
Thank you very much. Greetings from Vienna too. If there's any question, just drop me an email or give me a call.
Okay, thank you very much, Christian. We are now going to do the Kahoot dynamics. If you want to stay to watch it, you are very welcome.
Definitely, thank you.
Bueno, muchas gracias a todos. Vamos a pasar a la dinámica de Kahoot. Solo permítanme compartir la pantalla y mientras pueden ir alistando sus teléfonos celulares para participar. El pin para jugar está en su pantalla, 8219-724. Listo, vamos a iniciar. Gracias por participar. En un momento más les daremos un anuncio. Gracias.
Hola, buenos días. Nada más para informarles que tenemos un pequeño problema técnico. Vamos a retomar las pláticas en 20 minutos. Tómense este tiempo de descanso y nos vemos aquí a las 10:25. Muchas gracias.
¿Me pueden confirmar al chat si me escucho, por favor?
Hola, Ale, ¿sí te escuchas?
Ok, muchas gracias. Gracias. Bueno, pues es para mí un honor y un placer presentar a la maestra en Ciencias Rosa Arellano. Ella es bióloga de formación, tiene una maestría con especialidad en biología celular y actualmente es nuestra especialista aquí en Uniparts en el área de biología celular, con énfasis en cultivo celular en 3D y manejo de líquidos tanto manuales como automatizados. Entonces, le damos la bienvenida. Es tuyo el micrófono y la pantalla, Rose.
Buenos días. Buenos días, Ale. Muchas gracias por la presentación. Mi nombre es Rosa Arellano, soy parte del equipo de soporte científico en Uniparts y el día de hoy les voy a compartir una pequeña plática acerca de el manejo automatizado de líquidos usando Pipette Max para la detección de COVID. ¿Me pueden indicar si ven mi pantalla por el chat, por favor? Muy bien, muchas gracias. Les agradezco y una disculpa por los inconvenientes, pero tuvimos que desalojar un momento las oficinas, entonces ya estamos de vuelta. Bueno, el día de hoy vamos a mostrarles un poquito acerca de lo que es Uniparts. Me parece que estas diapositivas ya las vieron en la primera presentación del día de hoy. Y no se les olvide que al final de esta plática también tendremos un Kahoot, que muy probablemente lo vamos a realizar al finalizar la plática de Omega. Entonces, yo les voy a platicar un poquito acerca de este equipo que se llama Pipette Max. Es parte de Gilson. Ustedes, como saben, Gilson es una marca mundialmente reconocida, es de muy buena calidad y Gilson distribuye y fabrica materiales para el manejo de líquidos, desde micropipetas hasta estas plataformas totalmente automatizadas. Entonces, bueno, nosotros sabemos ya la pandemia que estamos viviendo. Al momento de tener esta pandemia, pues nosotros, toda la parte de la investigación pues se disparó, empezaron a investigar, se generaron las vacunas, reconocieron los antígenos, se generaron también, se montaron rápidamente pruebas de detección. ¿Y cuál fue una de las primeras pruebas de detección? Pues fue la de PCR, ¿no? Como bien sabemos. Al momento que surge esta pandemia o este virus y empieza a llegar a todo el mundo, pues nadie estaba preparado, ¿no? Los distribuidores, los fabricantes, nadie estaba preparado para la demanda mundial que provocó esta pandemia, ¿no? Entonces, por ejemplo, empezó en China, se empezaron a agotar los consumibles, los kits de extracción, empezó a haber una gran demanda para el diagnóstico de COVID. Entonces, pues todos empezaron a optar por máquinas automatizadas, porque sabemos muy bien que una persona en un laboratorio de diagnóstico que no estaban preparados, pues no había ni personal, ni reactivos, ni máquinas, ni equipos. Entonces, pues esto nos da una gran lección para poder estar preparados, tanto los fabricantes como los distribuidores, y también la parte de la investigación y todas las clínicas de detección, pues para poder enfrentar este tipo de retos, ¿no? Sin embargo, pues bueno, ahorita les voy a platicar un poquito acerca de esta parte. Yo sé que todos ustedes ya manejan toda esta información de cómo es la estructura del virus, ¿sí? Entonces, de manera global, ya sabemos que pues todavía no atravesamos por completo toda esta pandemia, sino que siguen incrementando los casos. Sabemos las nuevas variantes y pues todos estos diagnósticos se hacen por PCR, ¿no? PCR, ¿por qué? Porque pues es más sensible, ¿no? Hay otros tipos de pruebas como lo vimos, bueno, hay pruebas de antígenos, hay pruebas de anticuerpos, sin embargo, pues para la detección de COVID se recomienda el qPCR, ya que es muy, muy sensible, ¿no? Y es el gold standard para ahorita la detección y el diagnóstico de COVID. Entonces, ustedes sabrán que el protocolo que aprobó la CDC, pues es a base de hacer el qPCR, y pues aquí podemos ver una serie de pasos que es hacer, pues, primero tomar la muestra, obtener la muestra, extraer el RNA de la muestra, preparar los master mix, preparar el RT-PCR, ¿sí? Para poder hacer la detección y correr los ensayos, ¿no? Para esto nosotros necesitamos utilizar pues termocicladores para poder hacer la detección y el análisis de los datos. Entonces, como ya lo hemos visto a lo largo de estas jornadas, pues nosotros manejamos diferentes tipos de productos para poder hacer todo este flujo de trabajo. Como les comentaba, pues hay que hacer la extracción del RNA, hay que hacer la preparación de los master mix, alicuotearlos, transferir las muestras y transferir pues finalmente al termociclador para que nos puedan dar los resultados. Sin embargo, y como les comentaba, esta pandemia, pues de las muestras empezaron a incrementarse, ¿no? La cantidad de muestras que llegaban a los laboratorios de diagnóstico empezó a incrementar, y cada laboratorio, dependiendo de qué tan grande o qué tan pequeño era, pues podían llegar a tener hasta 5,000 muestras por día. Entonces, imagínense trabajar y procesar 5,000 muestras con una pipeta multicanal, pues es bastante trabajo, porque pues tenemos diferentes pasos que tenemos que hacer lavados, tenemos que poner reactivos, pasarlos a placas, transferir muestras, hacer alícuotas. Entonces, todo esto tarda mucho, es muy tardado si lo hacemos de manera manual. Nosotros sabemos que, por ejemplo, para poder hacer un PCR podemos tardarnos cierto tiempo, ¿no? Unas 2 horas, 3 horas, dependiendo de los equipos que manejemos, de los consumibles, pero al momento de tener tantas muestras, pues los resultados y el diagnóstico de la enfermedad, pues tardaba mucho más tiempo en dar el resultado. Entonces, al momento de que se incrementa la demanda y que empezamos a tener falta de personal, falta de consumibles, falta de todo en los laboratorios, pues se opta por utilizar máquinas, ¿no? Que nos puedan ayudar a hacer todos estos procesos que requieren de invertir tiempo, ¿no? Tiempo y esfuerzo. Entonces, aquí tenemos dos variantes del protocolo, de un solo paso o de 2 pasos, y sabemos que, por ejemplo, hacer 2 pasos en un protocolo, pues eso también te incrementa el tiempo. Todas estas pruebas de diagnóstico, pues como les digo, pueden hacerse dependiendo del termociclador en diferentes tipos de consumibles. Pueden ser en tubos de PCR o placas para PCR de 96 pozos o de 384. Entonces, si hablamos de 5,000 muestras diarias, pues vamos a necesitar muchísimos pozos. Y sabemos que el cansancio es un factor para cometer errores de pipeteo, ¿no? Ya que se ve afectada la precisión. Ahorita no les voy a platicar, bueno, no es un curso de pipeteo, pero si ustedes están interesados podemos hacer un curso de pipeteo en sus instalaciones, y vamos a darnos cuenta que tenemos muchos factores que pueden afectar al pipeteo, incluso cuando nosotros no lo hacemos de manera continua o frecuente. Cuando nosotros nada más alicoteamos algo, pues aprendemos, sabemos cómo pipetea, cómo nos enseñaron, pero a veces cometemos errores, y estos errores llevan pues a tener errores en los resultados directamente. Entonces, muchas veces también todos estos procesos, al ser manuales, pues puede que haya falsos positivos o falsos negativos por un único error de pipeteo. Entonces, es muy importante tener en mente que pues somos humanos y cometemos errores. Entonces, con el uso de estas máquinas o de este tipo de equipos, pues se ve reducido en gran cantidad, se ve reducida la tasa de errores que pueden llegar a cometer. Por ejemplo, les comentaba que pues podemos llegar a afectar la precisión, debido a que, bueno, la precisión pues es la capacidad de la micropipeta o el instrumento de dispensar cantidades parecidas de forma reproducible, es decir, que nosotros podamos poner la misma cantidad de líquido muchas veces. Aquí les pongo un ejemplito de lo que es exacto y preciso. Esto es lo que nosotros logramos usando una pipeta calibrada y con una buena técnica de pipeteo, evidentemente. Por ejemplo, acá tenemos algo que no es exacto ni es preciso. ¿Por qué? Porque la exactitud es la capacidad de la micropipeta o del instrumento para poder dispensar el líquido o la cantidad de líquido lo más cercana al volumen que nosotros queremos. Entonces, si nosotros le estamos marcando 10 microlitros, que sean 10 microlitros los que realmente estamos dispensando. Y la precisión es la repetibilidad, la forma reproducible, que esos 10 microlitros que estamos colocando los vamos a poder dispensar cada vez que usemos el instrumento. Entonces, aquí vemos que no es exacto ni es preciso porque no nos está dando los 10 microlitros y cada vez que lo hacemos nos da un volumen diferente. Aquí vemos esta parte que es preciso, pero no es exacto. Es preciso porque cada vez que nosotros pipeteamos el volumen es muy parecido. Sin embargo, no es exacto. ¿Por qué? Porque no nos está dando los 10 microlitros que le dijimos. Y bueno, aquí tenemos un ejemplo de lo que no es exacto ni es preciso, ¿no? Usando una pipeta o un equipo que no está calibrado y, por ejemplo, teniendo una mala técnica de pipeteo, que eso es lo que tenemos que tener en mente. Y como les decía, el cansancio es uno de los principales factores que nos lleva a cometer errores. No es lo mismo llegar al laboratorio, estar fresco, recién desayunado, con toda la frescura de la mañana, y empezar a pipetear, y después que no has comido, que ya estás cansado, ya no ves los pozos, pues empiezas a tener errores en el pipeteo. Eso también implica la pérdida de consumibles y pues de dinero. La automatización disminuye los errores humanos de pipeteo, también reduce la exposición a muestras infecciosas, y esto es un punto muy importante en este tema del coronavirus, porque pues nosotros sabemos que es muy alto, bueno, la tasa de contagio es muy alta. Entonces, con esto nosotros podemos reducir mucho la exposición de las personas que están trabajando con las muestras para que puedan llegar a contagiarse. También se reduce el tiempo de trabajo y se acelera la entrega de resultados, que es lo que queríamos y es lo que queremos en este tipo de situaciones. Además de que pues la reproducibilidad es una parte que es una cualidad que nos dan las máquinas, ¿no? Además de que pues evitamos todos los errores que podemos llegar a tener de manera manual, ¿no? Por ejemplo, que no embonen las puntas, que estemos cansados, que no dispensemos el volumen correcto, ya sea de muestra, de master mix, de cualquier otro tipo de reactivo. Entonces, Pipette Max es un equipo que tú programas el equipo para que haga, para que tome, dispense, mezcle incluso las muestras, únicamente dando play. Este equipo requiere de una computadora para que tú cargues el protocolo y entonces le des play y el equipo lo haga totalmente de manera independiente. Tú ya no tienes que estar ahí viendo y cuidando el equipo, y pues la máquina evidentemente no se cansa, puede trabajar prácticamente todo el día y no nada más sirve para hacer qPCRs. Este equipo es una plataforma abierta que te permite hacer ensayos celulares, ensayos enzimáticos, preparación de librerías para Ngs, manejo de líquidos en general y también, por ejemplo, protocolos de ELISA. Tenemos, por ejemplo, este equipo sin la cubierta se puede meter a una campana de extracción o una campana de flujo laminar para poder trabajar con muestras que no deban de contaminarse. Puede trabajar con puntas con filtro, puede trabajar en una mesa de laboratorio, es un equipo realmente pequeño. Todo esto el equipo lo hace mientras usted o mientras nosotros los investigadores nos concentramos en otras actividades. Nos da muchísimo más tiempo y producción, incrementa nuestra producción. Además de que este equipo es prácticamente un equipo de mesa, no es muy grande. Nosotros sabemos, y yo les comento que pues hay muchos equipos más complejos, pero que realmente abarcan 2 mesas de laboratorio. Obviamente son más complejos y por lo tanto son más cerrados. Son más cerrados a que tú no le puedes poner diferentes consumibles, no puedes usar otro tipo de puntas o no puedes usar placas de cierto proveedor. Este equipo es bastante amigable con todos los consumibles. Las puntas sí tienen que ser las específicas para poder asegurar la precisión y la exactitud, ya que todo esto se ve afectado por la par, ¿no? Por la pipeta y por la punta, que es algo que también vemos en nuestros cursos de pipeteo, que las puntas se recomienda que sean las que le quedan a la pipeta, porque muchas veces podemos tener puntas que son compatibles o puntas que son universales. Sin embargo, el que sea universal no quiere decir que haga un buen sello, y eso también nos puede llevar a errores de pipeteo. Entonces, este equipo es bastante amigable. No es tan complejo como las grandes máquinas de pipeteo que ya tienen incluso hasta brazos robóticos, pero el meter brazos robóticos o hacer más grande el equipo también complica la programación. Ya no puedes tú crear un protocolo diferente para ese equipo porque son muchas las variantes que tienes que manejar, que tienes que manipular. Se necesita de alguien o necesitarías tú solicitar un protocolo que te lo diseñen, pero pues sabemos que todo esto conlleva a cuestiones de programación. Aquí también se necesita, por ejemplo, el equipo tiene un software para controlar la máquina y tiene un asistente para hacer qPCR. Entonces, esto fue muy benéfico para esta pandemia, ya que con este programa de qPCR Assistant, tú puedes diseñar y crear tus propios protocolos para hacer el qPCR. Entonces, como es una plataforma que el cliente puede manejar directamente, pues estas máquinas empezaron a vender mucho en Estados Unidos. Y no nada más en Estados Unidos, también llegó aquí con nosotros toda esta tecnología y esta venta. Ahorita les voy a mostrar unos casos que tuvimos y que tenemos aquí en México, y en donde ya está trabajando este equipo para precisamente hacer el diagnóstico de COVID. Les comento también, no nada más se cierra el qPCR. Si ustedes tienen algún otro protocolo que quieran automatizar, por ejemplo, ELISAs, pues también nos pueden decir su protocolo si les conviene y se puede hacer una programación. Este equipo es un equipo abierto para poder hacer diferentes tipos de ensayos y utilizar diferentes tipos de materiales y de consumibles. Los equipos automatizados también nos permiten hacer ajustes para mejorar la precisión y la exactitud. Por ejemplo, elegir entre diferentes técnicas, ¿no? Nosotros sabemos que en las técnicas de pipeteo tenemos, por ejemplo, la técnica estándar, que es la que todos conocemos, pero los equipos de pipeteo también nos permiten elegir entre que hagan la técnica estándar o la técnica reversa, dependiendo del líquido o las características del líquido que vamos a manejar, porque eso es también algo que nosotros pasamos por alto y no nos ponemos a pensar en esa parte. Por ejemplo, la técnica reversa es conveniente para usar con sustancias viscosas o para evitar la formación de burbujas. También, por ejemplo, nos permite hacer ajustes de enjuagar 2 o 3 veces la punta con el líquido, que eso también es un tip que debemos de hacer cada que pipeteamos. Trabajar con cierto equilibrio de temperatura, porque aquí la temperatura ya no va a estar fluctuando, porque no va a haber un cuerpo, o sea, no vamos a estar nosotros directamente en contacto con el cuerpo de la pipeta. Ustedes saben que cuando tomamos la pipeta se calienta, y sabemos que el proceso de funcionamiento de la pipeta es mediante aire. Entonces, cuando calentamos ese aire, también nos lleva a tener ligeras variaciones en la cantidad de líquido que estamos dispensando. Aquí este tema de la temperatura pues no se ve afectado porque realmente el equipo está trabajando y este equipo no se calienta en esta parte de aquí o en la parte en donde están los cabezales. También algo que nosotros podemos decirle al equipo cómo lo haga, ¿no? Sumergir la punta a la profundidad correcta, que eso también es un pequeño problema que tenemos cuando hacemos pipeteo de manera manual, ya que muchas veces se nos olvida o lo queremos hacer muy rápido, entonces pipeteamos prácticamente a como a como lo vamos haciendo rápido, no nos damos cuenta de eso y también es un punto clave que tenemos que tener en mente. Y también, por ejemplo, podemos modificar la velocidad de aspiración y dispensado que mejor convenga dependiendo de las características del líquido que estamos manejando. Aquí, por ejemplo, les pongo una grafiquita en donde viene, por ejemplo, el error aleatorio contra, bueno, aquí viene el Pipette Max, un usuario con experiencia en pipeteo y un usuario con experiencia básica en pipeteo. Y este es el error aleatorio. Entonces vemos que el usuario con experiencia básica pues va a tener un error muy alto. De todas formas, tenemos aquí un usuario ya experto en el pipeteo y tiene pues tiene también error aleatorio. Sin embargo, vemos que usando Pipette Max, este error aleatorio pues disminuye en gran cantidad, en gran medida, ¿no? ¿Por qué? Porque podemos manipular y podemos controlar otros factores, otras variables, que cuando nosotros lo hacemos de manera manual, pues no nos concentramos en ellas, ¿no? Además de que pues los equipos nos ayudan a reducir el tiempo y el esfuerzo, incrementando la productividad. Por ejemplo, son las pantallas del qPCR Assistant, que es una parte del software que tiene el equipo Pipette Max. Como ustedes pueden ver, el equipo tiene aquí 2 cabezales. Puedes tú colocarle un cabezal de 8 canales para 20 microlitros y de 8 canales para 200 microlitros, y tiene una charola con 9 posiciones. El equipo se mueve en X, Y, Z. La charola tiene 9 posiciones en donde tú puedes colocar, bueno, este es el depósito para las puntas de desecho, los racks de puntas nuevas, por así decirlo. Podemos utilizar adaptadores para mantener frío o caliente tubos o placas. Son compatibles con placas de 96, de 384, con reservorios de 4, de un canal, de 12 o de 8. Ajá. También tenemos racks para tubos de 1.5, de 2 mililitros. Entonces, prácticamente tú aquí también puedes hacer muchos experimentos. Adicional a esto, si tú necesitas hacer extracción por perlas magnéticas, pues hay algunos aditamentos que te ayudan a hacer esos procesos que requieren algún imán para poder hacer la extracción. Entonces, es un equipo que es muy abierto, nos permite hacer muchas cosas y muchos tipos de protocolos. Les comentaba, este es el qPCR Assistant, es una parte accesoria del programa. El programa que controla la máquina se llama Trilution Micro, y con esta parte accesoria que es qPCR Assistant, tú puedes crear tu propio protocolo para tu qPCR. Puedes, por ejemplo, hacer y diseñar tu placa de qPCR, puedes hacer diluciones de las muestras y los master mix. Entonces, te va a llevar por una serie de pasos en donde tú vas a tener que decirle al programa cómo lo vas a hacer. Añades tus muestras, si tienes controles, si no tienes controles, tú diseñas primero tus templados y luego los cargas para poder asignarlos a las muestras. Aquí, por ejemplo, te dice cuáles son los datos del experimento para poder crearlo. Le pones aquí todos los datos y la información de tu protocolo. Te dan, por ejemplo, ya la disposición de las placas. Tú puedes cambiar el orden, lo puedes modificar, puedes ajustar también otros parámetros como lo son los tubos o los consumibles de donde tú vas a tomar los reactivos. Por ejemplo, si son tiras de 8 tubos, si son placas de 96, si son reservorios. Aquí vemos que ya vienen algunos precargados, y en caso de que no se encuentre algún consumible, pues nosotros podemos ayudar a diseñar un nuevo patrón para que podamos cargarlo en el equipo y bueno, sea compatible, ¿no? Aquí está la disposición de la charola, tú puedes moverla como más te convenga, y pues finalmente te dice dónde se van a guardar los resultados. Entonces, una vez teniendo eso, pues ya puedes tú hacer tu protocolo para diagnóstico de COVID o de algún otro tipo de enfermedad, ¿no? Entonces, estos son unos pequeños estudios de casos que hemos tenido. El caso 1 nos lo compartió Gilson directamente, que bueno, fue en un hospital de Estados Unidos, usaron el kit de Takara One Step para la detección de COVID-19 y lo que hacen con el equipo es preparar el master mix manualmente. Eso es lo que hacían, bueno, es lo que hacen, preparan el master mix manual y usan Pipette Max para licuatear y transferir las muestras. Entonces, con esto pues ya se ahorran toda esta parte. A veces hay hospitales en donde tienen un Pipette Max únicamente para los Master Mix y otro para todas las muestras. ¿Por qué? Porque se trata de reducir eso, ¿no? La contaminación cruzada. Entonces, eso también es algo importante que tenemos que tener en mente. El equipo lo puede hacer en un primer paso, que te haga la alícuota y que te haga la transferencia, y en otro segundo paso, cargar las muestras. Sin embargo, va a ser el mismo equipo que va a estar manejando los dos reactivos y eso también hay que tenerlo en mente. Estos son casos que hemos tenido, pero ya aquí nosotros directamente. Este caso es un Pipette Max que está instalado en el Instituto Nacional de Cardiología y aquí lo querían únicamente para la transferencia de muestras. Este es un caso que me gusta contarles porque es un poco cómico, ya que los investigadores del Instituto Nacional de Cardiología no querían el Pipette Max. No lo querían, pero se los compraron. Entonces, ellos querían una máquina más compleja, más grande, que tuviera cabezales que se podían, que era un cabezal, bueno, no era un cabezal, eran varios canales de las puntas que se podían abrir y cerrar, ¿no? Que no estaban los 8 canales fijos. Entonces, ellos querían otra tecnología mucho más compleja. Les compraron, su departamento de compras les hizo la compra del Pipette Max, porque lo que ellos querían hacer, Pipette Max lo podía resolver. Sin embargo, ellos no lo sabían. Entonces fue un problema ahí cuando llegó el equipo, lo echamos a andar, hablamos con ellos, diseñamos el protocolo y finalmente logramos lo que ellos querían, que era transferir las muestras para detección de SNPs. Ellos trabajan con SNPs. Entonces, lo que ellos querían únicamente era que el equipo les transfiriera las muestras de una placa de 96 a placas de 384, porque ellos tenían que llenar placas de 384 todos los días. Entonces, pues se tardaban muchísimo, ¿no? ¿Y a quién ponían a pipetear? Pues a los chicos de servicio social. Entonces, pues ahí volvemos a lo mismo. Tenían errores, había pozos sin cargar, había error de precisión y de exactitud. Si ponemos al chico inexperto, pues va a ser diferente al que ya tiene experiencia. Entonces, todo esto ya lo hace Pipette Max en estos momentos, ¿no? En el Instituto de Cardiología. Tenemos otro estudio de caso, que es un caso que este protocolo está en desarrollo. Este es un caso para el ISAS, el Instituto Nacional de Pediatría adquirió este equipo desde hace mucho tiempo, pero no lo habían echado a andar. Entonces, ahorita lo que quieren hacer es un protocolo de LISA. El protocolo de LISA ya lo tenemos desarrollado, ya lo tenemos, nosotros ya lo probamos, nada más estamos ahorita esperando porque pues como lo compraron desde hace mucho tiempo y no lo echaron a andar, pues tenemos ahí unos problemas de reconocimiento del equipo. Sin embargo, ellos, y ustedes saben que pues las ELISA son pasos que, por ejemplo, tienes que añadir el anticuerpo y luego tienes que lavarlo y tienen que hacer muchas placas de 96 pozos. Entonces, lo que estamos haciendo aquí es un protocolo para que pueda añadir todo el antígeno a todas las placas y luego todas las muestras y, por ejemplo, también curvas estándar. Entonces, todos estos protocolos ya los tenemos diseñados, ya nada más es cuestión de probarlos y hacer ajustes. Y finalmente tenemos un último caso. Este es el último caso que no es el último que hemos hecho, es el penúltimo, pero lo dejé al final porque les vamos a pasar un pequeño video acerca de lo que logramos. Entonces, este caso fue para la detección de COVID. El doctor Fernando Bastida, del Laboratorio Estatal de Salud Pública, adquirió el equipo para poder cargar sus placas porque le incrementó mucho la demanda del diagnóstico de COVID cuando llegó aquí a México. Entonces compró la máquina, compró el equipo, fuimos a instalarlo. Se tardó un poco en llegar porque pues ya había demanda en todo el mundo por este tipo de equipos y de consumibles. Sin embargo, cuando llegó, pues lo echamos a andar y el día de hoy pues está funcionando día a día el equipo. Entonces, este es el último caso que les comento y les vamos a pasar un pequeño video que espero que les guste mucho. Les voy a compartir mi pantalla. Aquí va el video. Denme un momento porque no se escucha mi video.
Inicié con la influenza AH1N1, ahí fueron mis primeros pininos. Inicialmente pues no teníamos equipo de termocicladores o equipos de parabiología molecular, todo se hacía por microscopía por campo oscuro, bueno, inmunofluorescencia. Entonces pues eran bastantes muestras y pocos ojos para leer tantas laminillas, aparte de que el proceso era muy tardado. Entonces cambiamos a la tecnología de punta, termocicladores en tiempo real, de algunas marcas reconocidas. Y ahí iniciamos nuestra carrera. En ese entonces pues no tenía ningún posgrado. Aprendimos a utilizar esta parte de estas herramientas de PCR a tiempo real. Y posteriormente, bueno, empezamos a investigar más, empezamos a meternos a posgrados. Y hasta ahora, la actualidad, ¿quién iba a decir que 10 años después una pandemia por coronavirus, ¿no? Pero bueno, ya para esta pandemia pues ya estamos más preparados. Ahora ya cuento con un doctorado y me especialicé mucho en la parte de PCR tiempo final, punto final, PCR tiempo real y PCR digital. Y bueno, aquí desempeñamos mucho las actividades, es 100% diagnóstico y un poco de investigación. Por ahí tenemos ya 5 artículos de SARS-CoV-2 que ya están acceso libre ahí en línea, si los pueden consultar. Y aparte de hacer el diagnóstico de SARS-CoV-2, también hacemos diagnóstico de chikungunya, Zika, dengue, campylobacter, SP, rotavirus, sarampión, rubiola. El motivo fue de automatizar todo, fue porque teníamos bastantes muestras, nos llovían muestras, había días que llegaban hasta 2,000, 3,000 muestras diarias. Entonces hacer esto manual era mucho tiempo, muy cansado, muy desgastante, y la otra es de que pues por las áreas pequeñas no podemos tener más personal. Nos topamos con esta máquina de Gilson, que nos ha ayudado bastante a cargar placas de 384 pozos. Un humano pues se tardaría cerca de 2 horas en hacerlo, y este equipo en 15 minutos ya está cargada la placa. Entonces nos ahorra tiempo, dinero y esfuerzo, ¿no? Bueno, esto nos tomó de sorpresa, ya que en el área solamente estábamos 2 personas, ya que los diagnósticos pues era influenza, dengue, Zika, que son por temporadas. Entonces esa parte, pues 2 personas lo sacamos bastante bien. Y cuando llegó esto, pues fue montar la técnica rapidísimo, o sea, de un día para otro, y empezar a buscar personal del mismo laboratorio que pudiera estar apoyando esta parte. Se contrató gente, inclusive llegaron algunos voluntarios, y fue algo difícil. Primero, la escasez de reactivos, puntas, consumibles, máquinas, y sobre todo, una vez que los conseguíamos, el personal, porque muchas cosas eran manuales. Entonces tener una carga de 1000 muestras diarias y hacerlo manual pues era maratónico. Entonces empezamos a buscar algunas máquinas y tratar de automatizar lo más que se pudo esta parte. bueno, no teníamos muchas máquinas. Inicialmente lo que pensábamos que hiciera este equipo era que nos cargaran los templados. Sin embargo, veíamos que cargar una placa de 384 de manera manual era mucho más tardado que cargar muestras. Entonces fue donde cambiamos el proceso y este equipo se ocupa ahora exclusivamente para cargar master mix de placas de 384 o de 96 pozos. Entonces, mientras dejamos el equipo procesando, nos da tiempo de hacer otras cosas. Bueno, pues aquí está la historia. Primero eran micropipetas, después llegó esta parte de la manual y pues por último la parte automatizada. Entonces, bueno, pues creo que está clara la historia. El tiempo, pues sí, una placa de 96, pues más o menos en 15-20 minutos. Después redujimos este tiempo a 10 minutos y posterior a 5 minutos. Entonces creo que la precisión y el tiempo se ve claramente, ¿no? Que se ha mejorado bastante, tanto tiempo y precisión. Porque bueno, como es una máquina automatizada, le pide unos 5 microlitros y 5 microlitros son homogéneos en cualquier pozo. Bueno, esto fue muy bueno porque ahora el problema ya no era la extracción, sino que una vez que llegaron las máquinas de extracción, el problema no era la extracción, sino que era el cargar el master mix. Tenía que tener una persona aquí exclusivamente. Y ahora con esta máquina, bueno, pues nada más es ponerle los reactivos y él se encarga de todo. Entonces lo que hacía una persona en una jornada en 8 horas, el equipo lo hace en 1 o 2 horas. Entonces nos ayuda muchísimo a reducir tiempos, mucho. Ahora con este equipo, no sé, ahorita hemos estado recibiendo un promedio de 700 muestras, entonces metemos una placa de 384 y el resto de 96, entonces esto pues en 1 hora ya tenemos cargadas las placas. Es muy amigable, realmente con el conocimiento así muy básico puedes programar la máquina y puedes ajustarlo a tus necesidades. Eso es algo muy bueno de esta máquina, de que tú le pones los volúmenes, cuántos master mix vas a ocupar, 1, 2 o 3, dependiendo lo que metas. Es un software muy amigable que yo pienso que cualquier persona lo puede manejar. No tienes que programar nada, no hay que hacer scripts, no hay que hacer otro tipo de ajustes. Eso es algo muy bueno y que pocos equipos hacen. Muchas veces, si no compras el consumible del proveedor, no puedes cargar tus muestras, tus reactivos. Entonces este equipo es muy abierto. Cualquier placa que le metamos, placa que carga. Esperemos que baje un poco más el trabajo, o más bien que nos llegue la nueva tecnología. Estamos en proceso de adquirir un secuenciador de nueva generación y queremos ponerlo a hacer las librerías. Pues la verdad estoy muy contento, redujo mucho tiempo y sobre todo la precisión. O sea, no hay de que muchas veces ya cuando lo hace uno manual y ya está uno cansado. Por ejemplo, cuando nada más teníamos equipos de 96, de placas de 96, y teníamos que cargar, no sé, 15 placas diarias, pues ya en las últimas ya uno ya está muy cansado, ya está uno harto de tantas muestras y todo, y algunas veces te llega a pasar cargar un pozo. En cambio en este equipo, pues no, desde la mañana ya sabemos cuántas muestras van a entrar, lo programamos, lo dejamos trabajando y podemos avanzar en otras cosas. Entonces, pues la verdad, mientras el equipo esté bien calibrado, esté con sus mantenimientos, no tenemos ningún problema de fallas. Ahora sí, aquí les puedo decir que del 100% de las muestras, de las corridas que hemos hecho, las 100% de las corridas que han salido, no hemos tenido ninguna falla.
Bueno, pues espero que les haya gustado mucho este video que hicimos con el doctor Fernando Bastida en el Laboratorio Estatal de Salud Pública en el Estado de México. Es uno de los laboratorios en donde más hemos trabajado para la detección de COVID y pues al parecer ya no tenemos mucho tiempo para las preguntas. Sin embargo, les comento que si tienen alguna duda, comentario o cualquier cosa me las pueden mandar al chat y nosotros les vamos a dar respuesta. Muchas gracias, les dejo a Ale Cavazos.
Muchas gracias, Rose, por tu participación. Sí tenemos algunas preguntas que le vamos a hacer llegar a la maestra en ciencia Rosa en el correo. Y por cuestiones del tiempo que perdimos, vamos a pasar directamente a nuestra dinámica de Kahoot. Les pido que por favor se conecten, que nos acompañen en este Kahoot. Recuerden que el ganador de cada uno de los Kahoots va a recibir un premio. Y para participar, por favor, ingresen el PIN del juego, que es 9446696. Muchas felicidades a los ganadores del Kahoot. Y bueno, voy a presentar a nuestra siguiente ponente, la doctora Eman Alexic. Ella es gerente de Producto Global en Omega Diagnostics Group. Estudió en la Universidad Monash y también tiene una maestría en ciencias, graduada con honores en el área de biología celular, con especialidad en inmunología. Tiene experiencia en investigación básica y diagnóstico molecular del VIH y actualmente trabaja en el campo del diagnóstico y salud global. Tiene también una amplia experiencia en programas de capacitación en una variedad de pruebas de CD4 y VIH. Y además es experta en sistemas de gestión de calidad para laboratorios de pruebas médicas de diagnóstico que se rigen bajo la ISO 15189. Entonces le damos la bienvenida. Les comento que le pueden escribir directamente sus preguntas aquí en el chat general a la doctora en español y su equipo va a estar respondiendo esas preguntas también en español. Nos las pueden enviar como han estado haciendo, o también si gustan al final de la plática desbloquear su audio y hacerle su micrófono, perdón, y hacerle la pregunta directamente, lo pueden hacer. Doctora Emma, we thank you for your participation in this event. We are very happy to have you here today. I hope that I have pronounced your name well. And the participants are already aware that they can write their questions in the chat or tell the questions directly to you. And at the end of the session, your work team will be answering in Spanish. I give you the microphone and the screen for your presentation. You are very welcome.
Thank you. I'm just going to put my screen on. I hope you can see this. Can you see my screen?
Yes.
Okay, great. Thank you so much. Gracias al equipo de Uniparts por invitarme a introducirles hoy nuestro producto. Mi nombre es Eman Alexic, como han dicho, y soy gestor de producto para Omega Diagnostics. Como pueden ver, hablo algo de español, pero no mucho. Y he hecho mi presentación en español para ustedes, aunque he decidido de hablar en inglés, lo siento. La presentación se la facilitaré a Uniparts para que la comparta con todos ustedes. Voy a introducir nuestro equipo, quienes son en la llamada hoy. Tenemos el grupo, tenemos Luz Molina, quien es nuestro formador de productos. Tenemos Neil Poynton, nuestro director de ventas para México, Latinoamérica, Asia y Pacífico. Tenemos Lorraine Nayoke, gestor de ventas de África Este, y Joseph Orton, nuestro director de ventas de África. Y John Bannister, nuestro director de desarrollo empresario. A lo largo de mi presentación, si tiene alguna pregunta, por favor, póngalas en el chat, y ya sea en español o en inglés. Luz es una hablante nativa de español, así que podemos abordarlas ahí. Gracias. Y ahora voy a cambiar a inglés. Okay, so the overall look at my presentation is kind of cut into two parts. The first part is looking at the general company overview and also the regulatory approvals and scientific evidence we have for the product. I'll also cover our funder and our stakeholder support, and then talk a little bit about CD4 and also advanced HIV disease for those who don't have that background. Then I'm going to then jump into the second half of the presentation, which is looking a bit closer at the product itself, the Visitech CD4 Advanced Disease Test, and our global partnerships, our commitment to training and education, and then there'll be some small time for questions and answers. So a little bit about the company itself. We're a publicly listed company and we're based in Elva in Scotland in the UK. We're a GMP site, so this means that we make our tests under certification of the ISO accreditation 13485, and we have over 30 years of experience manufacturing diagnostic tests. The test itself originated from an idea that came from the Burnet Institute in Melbourne, Australia, and actually I used to work for this company for a number of years. Since then, the Omega Diagnostics company has developed and now are manufacturing the product and have the rights commercially to distribute worldwide. We underwent successful tech transfer of the technology in 2015, and since then, we've had a period of optimization, verification, and validation, which has led to having this great product on the market. So this is the product itself. I'm not sure if you can still see me if I'm presenting, but this is the actual test. It's a lateral flow device. It's very small, like a pregnancy test or a HIV rapid test. It has a special marking which indicates a level of CD4 that's at 200, so the patient result will be above or below a 200 cutoff point. And the test itself is CE marked. It was CE marked in 2019, and we have a number of different approvals, and I'm just going to go through those now. The second on the list is ERPD, which is a global fund approval, which we received in September 2019. The third one on the list is an EMAV, which is a signed agreement with a company, a global organization called Unitaid and another company, Chai. And we signed this agreement in April of 2020. This is for short called EMAV, but it's basically an early market access vehicle, so a way for the product to enter the market at a very small price or for free. And I'll talk about that a little bit later in my presentation. The last but not least point that I want to talk about is that we have WHO pre-qualification, and we received this qualification in September 2020. So just to highlight, there are two very important points here. The Visitec CD4 disease, advanced disease test is the first and only rapid test that's semi-quantitative on the market, and we're the only rapid test that has WHO pre-qualification. Okay, so a little bit about the performance results that we have for the product. At the very top,
Creo que la doctora Eman tuvo un detalle ahí con su conexión. Vamos a esperar por favor unos minutitos a ver si se vuelve a conectar.
hello, can you hear me again?
Yes, we can hear you again, doctora.
Okay,
But
thank
your
you.
screen, we cannot see your screen, your
Yes,
presentation.
I will share that in one moment. I'm sorry, I'm not sure why it keeps cutting me out. Can you see my screen now?
Yes, we can see it.
Okay, just make it a slideshow. Okay, I'm not sure where we got to last time. Did you see this slide?
I think one before.
Here?
Yes.
Okay, so just quickly, I'll just make some highlights here. I'm not sure what you did see, but just to highlight that the test itself is the first of its kind. That's a rapid semi-quantitative CD4 test, and that it is the only test that is WHO pre-qualification. Okay, so just to continue, this is some data from our studies that we've done on the product. At the very top, you can see that we've got the results that are inserted into our product insert for the performance evaluation. You can see that we have the sensitivity and the specificity is very high, whether you're using a capillary sample or a venous sample. And then below this, you have data that's been published from Doctors Without Borders, and this shows also similar data that's been now published in PLOS One in 2020. Okay, so next I'm going to move on to talk about some funding and stakeholder support that we have. So at the first of the list, we have the COP guidance document. This is a document that outlines what countries will be able to purchase or look to purchase for their countries for the year ahead. And in the COP21 guidance, PEPFAR, who write this guidance, listed the Visitech CD4 Advanced Disease Test in this guidance as a new point-of-care device to support patients living with advanced HIV disease. Second on the list, we have CHI, who at their mid-year report of this year also mentioned Visitech CD4 Advanced Disease, and it's the EMAV that I mentioned earlier on my slides. So next, going into the EMAV itself, I wanted to just talk about what this means. The Unitaid CHI Early Market Access Vehicle is a catalyst. It's a way to encourage ministries of health in countries to introduce the Advanced HIV Disease Package of Care. And we've already started work in Nigeria, Uganda, Zimbabwe, and we're now expanding into Mozambique, Central African Republic, Guinea, Democratic Republic of Congo, and South Africa, and hopefully soon to countries in Latin America. If anyone has a use for this product locally, we would really encourage you to look into this a little bit more in more detail, and we're more than happy to answer questions about this. What this really means for you is that you can have access to this test through a coordinated mechanism that allows you to get access to the test without any costs associated. And this is covering over 120 countries and includes Mexico in this list. There are some things that you have to think about in terms of this, but the main detail to note is that this has expressions of interest that are open until the end of September this year. So there's still time to learn more about this and find more information. And as I said, we will share these slides. So you'll be able to find out more information through these links that are on this slide, or we can provide them to you directly. Okay, so finally about what it is and why we talk about CD4 T cells. For those of you who don't have this background or don't remember, this is just a little bit of information about that. So we talk about CD4 T cells being the reason, an indicator of a HIV positive patient's immune system during their infection. And so what I would like to do initially is focus on this image on the left here. And in the first test tube, these are all sort of schematics that represent a patient's blood as they're undergoing, as they've, pre-infection and then post-infection phase. So you can see that in the first test tube, there's a lot of blue circles. These circles represent CD4 T cells. And this is pre-infection. Post-infection, you can see that there are a lot of red dots, and these are representations of the virus particles. So we know in the weeks to months that follow infection, we call this the acute HIV infection phase, that there are virus particles that are attacking the CD4 T cells, and what happens is they attack, they adhere, and then they insert themselves, they replicate, and when they expel replicated, they kill the CD4 T cells. So this is the process of HIV. And over time, in the years following infection, we call this the chronic HIV infection phase. You can see that the number of CD4 T cells is starting to decline, and the number of virus particles increases. Finally, you get to this last stage, which is advanced HIV disease. In this stage, we call this advanced HIV disease because a lot of our HIV positive patients are now living, many years following infection. In the old days, we would call this AIDS, or in Spanish, we say SIDA. So we know a lot about this group, this last group, this advanced HIV disease group. We know these things. These are the 3 top points that I'd like to point out. First is that 1 in 3 patients are still, to this day, in 2021, presenting with advanced HIV disease at their first visit to the clinic. The second point I'd like to raise is that the WHO recommends a test for CD4 to identify advanced HIV disease in patients. There's no other marker for this at the moment. And the third thing is that when we know a patient has advanced HIV disease, we know that this can lead to opportunistic infections like cryptococcal meningitis, tuberculosis, toxoplasmosis, pneumonia, and histoplasmosis. So what this means is that we need to then start looking at addressing that once we've identified advanced HIV disease in patients. The next point I'd like to talk about is the current standard, the gold standard technology, or the reference technology, which is currently used to detect the level of CD4 in patients. We have the flow cytometry technologies. We know a lot about that, of course. It's been around for a long time. It's in a lot of the in a lot of countries, there's very limited infrastructure and money to support this technology. And we know that because of these constraints, that a product like Visitech CD4 Advanced Disease would be very useful. Next, I'd like to talk about the WHO definition for advanced HIV disease. So on your screen now is from the guidance document for managing advanced disease from the WHO, and I'm just going to talk about the first point. This is the definition of identification of advanced disease, which determines a patient with advanced disease with a CD4 count that is below or equal to 200 cells per microliter, or they have a WHO clinical stage 3 or 4 event. Okay, here we then are moving on to take a closer look at some of the barriers to access CD4 testing. So things that could act as a barrier include, and they're not isolated to these on this list, but could include viral load testing. So some countries have taken viral load testing on and dropped CD4 testing, but as we know, they're not always linked, and so this can be a misleading representation of advanced disease in the community. Sometimes the equipment that you need for CD4 is not available, and of course COVID has affected a lot of patients being tested for CD4 because some of the machines that are being used are reallocated to COVID. Also, we know a lot about where people live. In a lot of low to middle income countries, patients don't live very close to healthcare clinics, and many live rurally without access to care. And those patients who live in those rural communities may have to take blood in a different location or ship their sample to a different location. And sometimes they'll need more than one visit to get their result. And of course, on the right-hand side of your screens here, you can see that there is a diagram. This diagram talks about the patient pathway. On the left-hand side in gray, we have the conventional pathway. So this is what happens typically now in a country. A patient will come for care, blood is sent to a lab for CD4 testing. The patient then will have to wait maybe 1 to 2 weeks for a result, and then the patient returns to the healthcare facility for their clinical management. Okay, so this takes a long time. With this CD4 advanced disease test, what we see is that the patient's blood can be tested instantaneously and the clinical management can happen in the same day. So what are some other benefits for CD4 testing? We can see here that it not only is advantageous for the person living with HIV, but also helpful for the healthcare workers themselves. Again, reminding you it's the first test that doesn't require a machine in the world, and it's a very simple solution to evaluate CD4 levels. So here are our sort of top 5 features. The first one is that it really helps accelerate management of patients in the clinics, and it helps reduce the burden on the healthcare system itself because we take decisions faster. The second point is that we reduce the patient loss to follow-up. So this means when a patient comes to the clinic but then isn't able to be retained in the system because of the fact that they have to come for a second visit. And third, we say that it reduces costs because you don't have any equipment costs and you don't have to transport the test or the samples anywhere to be tested. The fourth on the list is just talking about testing anywhere, anytime, and that it's a very convenient, easy to read, read by eye result. And last but not least, and this is the most important, is that you get your results quicker to the patient. Patients with advanced disease can be detected earlier and then investigated for opportunistic infections faster. Okay, so having a closer look at the kit itself, this on the right-hand side is what the kit looks like when it's opened up. And you can see that there are a number of things included in the kit. We have the instruction for use, we have a job aid over here, we have a buffer bottle, and we have 25 devices. Each device comes in its own pouch with a desiccant, and then there's also 25 alcohol swabs, 25 lancets, and 25 capillary sampling devices. In the second group here, we can see that there are some materials that are not included, things like biohazard bins, gloves, timer, a pen. These are all things that are generally found in laboratories. And of course, if there are items that you would need specifically, for example, you would need a pipette if you're taking venous blood samples and adding them to the device. So there's 2 different ways that you can use the test. So that just leads me to talk about the actual test procedure. As I mentioned, you can use either a capillary or a venous blood sample. What we need to do is obtain your sample via either method, and then you add 30 microliters of your sample into the first well A. And that's right, it's 30 microliters. So it's just a drop of blood that's taken from the patient sample. Then there are a few simple steps to follow. So we have to then wait 3 minutes, and then we add 1 drop of buffer to the same well A. Then we have another wait of 17 minutes, which is followed by adding 3 drops of buffer to well B. And then we have a 20-minute wait, and then after this time, you can read the result within 5 minutes. So really, we say that it's a simple test, but there's a lot of complex chemistry that's happening inside. And I will go into the detail of why we have these important timing steps and why we add the buffer in this kind of way in a few slides from now. But first, I want to show you what it looks like to read a result. So first of all, to learn about this, you need to then have a look at what is a valid result. So on the left-hand side of your screen, I'm going to talk about this first. When we finish the test, we've got 5 minutes to read the result. And the first things that we need to look for are that we have a C or a control line, we have a reference line, and for this case here, we do have a T line, but the most important part is the línea de control and the línea de referencia. Okay, so these 2 must be present for them to be for the test to be considered valid. And on the right-hand side, you can see that there's no línea de control and there's no línea de referencia, so we need to repeat the test. Okay. Next, we're looking at what happens if a result is above reference. So this is again a valid result because we have a C line and we have a 200 line or the reference line. we know that if the T line, and now we're going to focus on T line, if this line is darker in color than the reference line, then we would say that the result is above reference or encima de la referencia. Okay, so patients with this kind of result would have then a greater than 200 CD4 T cell count. And we also want to associate this with a strong immune system. If we look at results that are below reference, and here you can see an imagine of this in images that could present this. So in all three of these images, you can see that there is a reference line and a control line. So these are all valid results. And then third, we're looking at the T line. Is that T line darker or lighter than that reference line? And in all of the cases here, this one it's equal to in color, this one is lighter in color, and this one the line is missing. And in all of these cases, the result is equal to or below 200 CD4 T cells, or we would say it's debajo de la referencia. And what does that mean? That means that the patient's immune system is weak and that some form of action needs to be taken. Okay, I'm just going to show a short video. I'm not sure if this will work with the sound, but please do let me know if you cannot hear, and I'll stop the video and keep going with the presentation.
Busy tech keyword for this own instrument-free rapid test on the market, support identification of advanced in people living with HIV. How to perform the visited C4 fast disease test. Materials provided to run tests on Enterotype, complex, bottle buffer, and alcohol swab to blend it. Instructions to use a job are provided in each kit box. You will also need disposable gloves, type of hair, a sharp skin, and trickles or tissue. BDTA with sampling will need an EDTA like culture tube, sympathetic and disposable. Always refer to the instructions you for the most up-to-date version of the test procedure. Allow the device and components to current rated temperature of between 50 and 35 degrees Celsius before use. Do not open the full pouch until you're ready to perform the test. 12 weeks before the test, check foil pouch and image. Check the date on foil pouch and components are intact. Tear open the foil pouch and remove materials, and you're ready to continue to this. If there's present, decide to use other device. Dispose of packing in a general extent. Write a patient identifier test on new air disposal plus. There are two types of cells, capillary and venous EDTA. You will cover capillary first. Page, which is a non-dominant inside of the figure with the alcohol of will be formed. Other things to air dry. Twist off on scan. Kiss the skin of the tip, side, or the finger. Dispose the retardants into jar bin immediately. Or wait for a drop, like a piece of rock or tissue, and dispose of in the jar bin. Gently squeeze the finger and full drop blood. Hold on the device horizontal and the tip of the arm moves the blood arm. Capillary angle will draw blood line, which is 30 microliters. To squeeze the hole to draw blood into the tube. Use the swab immediately. We'll now cover venous EDTA blood celling. Collect the sample using stash tape. For the position volume 30 microliters as chosen. EDTA blood outline gel infusion at 10 times. Binge for leg EDTA blood tube. Press the lunge button open enter the first time. Immerse the both tip vertically the EDTA tube. Simply release the tube, pouring blood in disposable tip. Whatever sample you use, capillary or venous, the rest of the test press is the same. Just then, weigh like and squeeze the valve of the arm device to the gently 20 to 30 release released until the end. disorders will take 8 3 minutes. After add 1 drop of buffer to 1 egg. Hold the bottle at a 90-degree perpendicular to this device. 1 centimeter well egg. Do not squeeze the bottle too hard. Wait for 17 minutes to 17 minutes pass at 3 drop buffer to LP. Hold buffer bottle to 90-degree angle to the device. 1 centimeter above the do squeeze the buffer bottle too hard. 20 minutes. After the test complete, invert results within 5 minutes. It's extremely important to know how to correct the test. To determine the test is valid, both control season and reference undefined must present reading the truths. Results are indicated visually by comparing all against the test line, the reference undefined. Test C line is the capacity as reference to 100 line. The CD count is estimated to be equal or low to 200 cells per test as bonus. If the test line lighter the reference to 100 line, the CD count is estimated to be less than 50 cells per program. The test is as below reference. Note that if the test line invariant or missing, the CD count estimated to below 284 cells per microliter, and the cell is interpreted as below reference. If this time marking intensity than reference to 200, CD4 count is estimated to be greater than 200 CD4 cells per microliter, but it is interpreted as above reference. If the reference line is missing, test results is repeat test. Control C line is saying the test result is invalid. The test you have finished interpreting the test by those domestic gloves in shopping. Visit CD4 assisted supporting the identification of advanced disease in all living HIV. It also
Okay, so that was just a short video about the test, just to go over the test steps, very high level. Now I'd like to talk about the test procedure in more detail, why we have the incubation steps that we have, and why we add the blood and the buffer drops in that specific order. So the first thing I'd like to say is that we're talking about whole blood that we're adding to the test device. And we know that whole blood contains a number of different components. We have red blood cells, we have monocytes that express CD4, and we have T cells. And when we first take that first 30 microliter sample of whole blood from the patient, whether it was capillary or a venous sample, we add that to well A. And in that first 3-minute incubation, what happens is the monocytes and the red blood cells are kind of taken out of the equation. And we're just left with T cells, or the CD4 T cells, and the plasma that can then pass through the membrane. The second step, which is talking about the addition of the buffer, the 1 drop of buffer in the same place as the blood in well A. In the first this step we call the liberation of the CD4, or the full-length CD4. And in this step, what we have here on the left of your screen is a schematic of a T cell. And you can see that the different CD4 proteins are visible on the surface of the cell membrane. In this step, when we add the 1 drop of buffer, what happens is that there's a lysis step that occurs. And now we have full-length CD4 that has both the intracellular and extracellular domains exposed. So after this first drop of buffer, we then are told to wait 17 minutes. And the reason why we have to wait 17 minutes is so that the CD4 protein can then run up the membrane. And so here what we have on your screen is a schematic on the side of this result window here. So here you have the C line, the reference line, and the T line. the first line here, see that there's the patient line, and now we have the intracellular domain of CD4 binding to a capture antibody that's already been bound on this membrane. And of course, in some patients, they may not have as many CD4 cells as another patient, so we need to wait the full 17 minutes to make sure it's accurate enough to capture all of the CD4 in that patient. In the next step, in the third step, we're looking at the visualization of the CD4. So all of the hard work has actually been done prior to this step, but now we're labeling with our gold conjugate. So when we add 3 drops of buffer to LB, it's really just to liberate that gold conjugate. And once that's liberated, it can now bind to wherever there was CD4 bound on the patient line. There's a second gold conjugate that will then bind to the reference and the control line, and these are non-competing, so we can see all the lines. Okay, so that was just a little bit about how the test works. I hope that was clear. Please ask questions in the chat if it wasn't. Next, I'm going to move on to talk about some of our global collaborations and networks that we've developed over the past number of years. Since our WHO pre-qualification, we've expanded to conduct a number of projects globally, and over half of our projects are using an advanced HIV disease package of care. And so we see the absolute need for a test like this right now. I'm going to highlight some of our projects in a moment, but before that, I will go into where we're covered right now. So at the moment, we have got projects that are in a number of different levels of the healthcare system. We've conducted over 55 training events worldwide with over 300 end users and trainers. We are in over 40 different locations, and we covered 25 countries around the world to date. Okay, on the right-hand side, you can see, on the left, you could see the world map, and this is where some of our projects are. And then on the right-hand side, you can see the distribution of some of our projects and where we've worked to date. So you can see a majority of them are in sub-Saharan Africa, but we do have a few projects in the Latin American Caribbean region, and I will talk about them today. So we have a number of objectives, really, that we want to continue to expand our coverage with this product and conduct more training events so people can learn about the product. And we want to really empower our country teams to become trainers and local end users and champions to continue those rollouts. We recently obtained registration in Brazil with Anvisa, and there's definitely a lot of support for the product in the region. Okay. So one project that I will talk about again is the Médicos Sin Fronteras project, or Doctors Without Borders project that has been published. You can see this project was 2 phases. It covered 3 countries, Democratic Republic of Congo, Malawi, and Zimbabwe, and it had over 1,000 patients recruited. The data that you can see on the screen below is data that's comparing to flow cytometry. So using that reference method, I know there's been a couple of questions about comparisons to the reference method. So all of this data is direct comparison. So you can see here in this example for capillary or for the venous, that there is over 5% sensitivity, which means the correlation to flow cytometry is very high. So just to highlight that point. On this slide, I'm just going to talk a little bit about a project that has looked at specifically the advanced HIV package of care. I apologize that this slide is not in Spanish, but I really want to focus in on this and the page here. Map. This is Map Senegal. The first phase was conducted in Dakar, and this is now being complete. And I'll show you the data on the next slides. The second phase is about to commence, and it's happening outside of the Dakar Center. It's happening down here in this region. okay? And they're looking to recruit another 500 patients in that region. And what they're planning to do is summarized in this table, where you have sort of the algorithm of care if you're looking at advanced HIV disease. So if you look at the very top, it says if the patient has a less than or equal to 200 count or a clinical stage 3 or 4 event, that patient would then undergo the testing pathway for testing for TB or for cryptococcal meningitis and so forth. That's the pathway. So this is something that we'd like to replicate globally. So looking at the phase 1 data, again, this is the data that compares our test directly to, in this case, the BD fax count machine. And so you can see on in this table down here, the sensitivity specificity is showing very, very high, greater than 90% sensitivity and specificity for this first phase of the project, which recruited over 200 patients. We also conducted a study in the Dominican Republic, and it was run by the National Reference Laboratory there, and the data speaks for itself. They saw 100% sensitivity and 100% specificity in those patients recruited. And in that case, there was 200 patients in that study. And at the moment, we're conducting another project in Argentina with Hospital Fernandez in Buenos Aires, which is looking at comparing the Visitec CD4 advanced disease test to the BD fax caliber machine, and then follow-up testing with the determined TB LAM test, Fuji LAM film test, and CRAG or cryptococcal meningitis test, and a histo test. And in this project, they'll be recruiting 200 patients. So this project has started, but we don't have any data to share yet. So I'd like to now jump over to discuss a little bit more about our support and our, what we'd like to provide as our compromiso to education and ongoing support. So we do provide a unique program. We can tailor a best fit solution to your needs. Our group consists of a dedicated product application trainer, and that is Luz Molina, who's on the call today, to develop and adapt a program that will suit your needs. Then we have also a group that works at our head office that can be waiting for your technical questions. And then, of course, we have local distributors. So as you have Uniparts in Mexico, they are trained to answer questions on our product and also obviously help you with whatever support you need to get this product launched in your facility. Second, I'd like to focus some energy on the kind of resources that we can provide to you. We have a number of materials in English, and in your case, it would be more important that we have a lot of materials actually in Spanish. We can conduct the trainings virtually through Teams, Zoom, or using medias like WhatsApp. And we've done a lot of that since the COVID pandemic happened. We have in-person training where this is possible, and we have a number of support materials that can help you understand and also teach the product. And recently, we introduced a global learning center. This is an online learning program which you can access online. So I'm going to show you a short video about that in a second. But first, I'm going to show you a couple of our materials. So, of course, we have the product insert that comes inside the kit, and then we have an ayuda de trabajo or a job guide. These are really handy. One side is for capillary, one side is focused on venous testing, and you can use these to sort of have a short step-by-step guide on how to do the test and read the result. And then we have a number of different support materials that we can provide. So what is the relevance of CD4 testing? How do we interpret sensitivity and specificity, and other technical guidance documents like how do you take a finger prick sample and how do you interpret results? So this is just an example of one of those such guides here. And of course, we have the video that I showed, and we have a lot of FAQs, so frequently asked questions, and a training manual. So next, I'm just going to talk briefly about our GLC, or our online learning program, and this I hope you will be able to. Okay, so that short video just sort of described a little bit about what the Global Learning Center is and what you'll get access to. So it's purely online. You'll get a login once you've been requested and given access, and this is your login screen. Then on the next image that you'll see, this is just talking about what is covered in this online program. So it has a course and an evaluation. So there are 4 different modules, and you can see these modules that are listed here on your screen. There's a welcome and introduction. We talk about the key product features, what to do before you start testing, how to perform a test, and then finally you'll have an interpretation quiz. It's estimated to take about an hour to complete from start to finish, and each section is blocked. So you have to complete section 1, module 1, before you can move on through the course. There's, at the very end, the interpretation quiz or the evaluation is a 50-question evaluation, and you need to get 95% to pass, and then you'll end up being able to download a certificate at the end. Okay, so in terms of the evaluation itself, I'm just going to show you a little bit about this. I'm not sure if it will let me navigate. It may not, so I might just move on. But basically, the interpretation quiz is a number of different examples of the how to read a result, and it takes you through that, so you get a lot of practice. So I know that a few people have questioned, how do you read a result? How do you become confident in that? Well, here in this interpretation quiz, we have real life images of the test, and you can see that and practice for yourself. Okay, so in conclusion, I just wanted to say a number of key take-home messages. That we know that the Visitech CD4 Advanced Disease Test is really a gateway test to the Advanced HIV Disease Package of Care. We are very pleased that we are WHO pre-qualified, and this opens a lot of doors in order to get funding for this product. So please ask us questions about how you could access this test that way. We have the CHI Unit 8 agreement that we talked about. This is that EMAV that I spoke of, and that there's still time to make applications if you're interested to put in a project, so please contact us about that. We have a number of organizations that are really providing a lot of support for this product at the moment, and we have a lot of scientific evidence that's supporting the Ministry of Health adoption of this product. So I just want to also finally end on the last 2 points, is that, you know, in terms of the support for trainings, for implementation, and for scale up locally, you know, you have us as the Amiga Diagnostics team and Uniparts to help support that. And we also have this really great online tool that can help you learn more about the product. So with that, I'll just leave you on this slide and ask if there's any questions that we might be able to answer. Thank you so much. I hope that there weren't too many technical difficulties with my presentation.
Thank you, Dr. Ayman, for your participation. The questions were already written in the chat, and the Dr. Luz Molina helped us answering. I think there are no more questions for you.
Oh, okay, perfect. Thank you so much.
Thank you, Dr. Ayman. And we are now going to the Kahoot Dynamics. If you want to stay to watch it, you are very welcome.
Yes. Thank you so much.
Bueno, muchas gracias.